Yep. Hi, everyone. So let me do a quick introduction of Rina. Rina is a first-year graduate student in bioengineering and in the medical imaging informatics track. She comes to us graduating from with cum laude from Case Western University, Reserve University in Cleveland, Ohio. And she did her bachelor's in computer science and the AI track and has worked at several AI labs, including the Center for Computational Imaging and Personalized Diagnostics. So today she will be talking about a project that we're looking at in terms of combining multiple pieces of information from imaging. As many of you know, with magnetic resonance imaging, you can get a lot of different functional and anatomical information. And so she'll be talking a little bit about how to combine that with the goal of really understanding ultimately the biology of the disease that we're imaging. So with that, Rina, why don't you take it away?
Yeah, thank you so much, Dr. Xu, for the introduction. Yeah, so like Dr. Xu mentioned, I have a computer science background, and today I'm gonna share with you our current work in the domain of multimodal data integration in prostate cancer. So even though the data type we're talking about today is imaging, I believe that the technique that I'm gonna introduce is really gonna be broadly applicable to other data modalities as well. So I'll be talking about the concept of multimodal data fusion first, and then I will talk about our work in prostate cancer, and then conclude the talk. So as we know, there's like increasingly abundant amount of biomedical data with different modalities, such as omics and genomic and imaging data. You can see in this figure. And each of those data capture a slightly different information about the disease. And when we combine them, they might give us a fuller picture of the disease. So the goal of multimodal data fusion is really to integrate those modalities together so that the model might have a better performance in both diagnosis and outcome predictions. However, in order to computationally model those multimodal data, we have to overcome some challenges. So in this picture, I'm showing you the idea of multimodal data fusion in prostate cancer. As you can already see, these different types of data have very different dimensionality, scale, and format. So then how do we overcome them? And in the case of wanting to fuse different imaging modality together, we have to consider how you actually spatially align them. So for example, if we want to fuse the pathology image with the MRI, we have to consider a point, which is that for pathology specimens, they're taken out of the human body, which means that they might deform. And that makes it harder to align with other imaging modalities. So then once we align those images or fuse other data, and we found some associations between them, then how do we actually validate them? And the last challenge is that since we have many different modalities with different noises and missingness, then how do we actually weight them differently? And in our work currently, we're focusing on the first aspect, where we used a dimensionality reduction-based data fusion technique, which I'll talk about later. So a little background in prostate cancer. It's the second leading cause of cancer death in men in the US. And the primary treatment is the radical prostatectomy, where the surgeons take out of the prostate from the patient. However, a great amount of those patients experience what's called biochemical recurrence, which is defined by a biomarker called prostate-specific antigen. And actually, around 2/3 of those patients have developed the BCR after around first 2 years of the surgery. And the BCR actually can increase the risk of metastasis and death, which is not good. And therefore, it's very important to identify those who are likely to develop BCR so that we can see if the surgery is a good option for them, and also provide some basis for determining who will need further treatment. And one way to identify abnormalities is to look at the multiparametric MRI. And in the MRIs, they can provide the image-guided biopsies to sample and then confirm the tumors in pathology. And to give you an idea of what those images look like, here I'm showing you a patient with clinically significant prostate cancer. And we have the MRI and the corresponding histological specimen. So for T2-weighted MRI, it's used as a primary diagnostic tool in a clinical setting because it has a high resolution for visualizing the tumor structural or anatomical information. And in the diffusion-weighted imaging, it evaluates the motion of free water molecules within a tissue. So then how does that tell us something about the tumor? Well, usually when there's dense cellular structure, they would impede the water molecule diffusion. So then when we have a tumor with high cellularity, then that is going to be quantitatively reflected on what's called the apparent diffusion coefficient map. And usually when there's a tumor, the ADC value is going to be lower. And as you can see at this region that captures tumor, it looks slightly darker as compared to the other regions. And as you can see, these different types of imaging provided slightly different information about the tumor. And in this study, we would like to combine those modalities together in order to predict which patients are more likely going to develop biochemical recurrence. So in terms of how to fuse the data together, there are two broader categories of techniques. One is early fusion, where we have, say, two modalities, and we want to transform them into a common space where there's the fused data. And then we use a single model to do the prediction on that fused data. And in late fusion, we have two separate models that make two separate predictions based on each modality. And then at this final step, we fuse their decisions together. So as you can see, in the late fusion model, we actually cannot capture the inherent relationship between the two modalities. However, the early fusion can. So then that's why we choose to use early fusion technique in our paper. And for early fusion, we have two categories of techniques, which is deep learning based and then dimensionality reduction based. And we chose the dimensionality reduction based technique because it's a more interpretable technique and also requires less amount of training data. And one commonly used technique is called canonical correlation analysis. And it's actually widely used in neuroimaging, but not as much in the other disease domains. Yeah, so finally, our goal in this study is that we want to extract quantitative imaging features, which are called radiomic features, from both T2 and the ADC maps. And then we want to fuse them together using a variant of canonical correlation analysis, which is called Sparse, SCCA. And then we want to use them to predict the likelihood of a patient having BCR. So the hypothesis is that the SCCA fused model can perform better in predicting BCR as compared to a series of baseline models, such as the ones that only use radiomic features from T2 and only from ADC, as well as when you just directly take those two feature matrices and concatenate them together. And as well as the clinical assessments of T2 score, T2 shape, and T2 signals. And these are qualitative assessments made by the radiologist, as well as the average ADC values. So for the data set, we use a UCLA cohort with a total of 470 8 cases with annotations. And then we have some inclusion criteria, which are that the patients had to have at least one post-surgery prostate-specific antigen value, and also have at least one biopsy-proven lesion. And they also have to have both T2 and ADC images and the clinical assessments that I mentioned earlier. So then eventually we had 343 cases, and we randomly subsampled 70% of those cases to be in the development set and 30% in the independent test set. And in total, we have 70 patients who developed BCR, and the median follow-up for them is around 12 months. Here's an overview of our method. Firstly, we extract the radiomic features from the annotated regions, and then we use the SCCA to fuse them together. And then we develop a prognostic model to predict the biochemical recurrence-free survival by the Cox proportional hazard ratio regression using a lasso feature selection. And then we used a 5-fold crossfade validation to choose the regularization parameter. And then we evaluated the model performance using Kaplan-Meier analysis and some other metrics. And at the end, we also investigated the contribution of the radiomic features to the fused representation. So as for radiomic feature extraction, we firstly did some pre-processing on the images. And then we took the annotations by the radiologist at a per 2D slice level, and then render them together so that we get a 3D view of the lesion. And as you can see, the lesion or the tumor is the red region, and then the green is the prostate. So then we extract different features from the region, such as shape, intensity distribution, and texture. So the shape and intensity distributions are relatively intuitive to understand. And now I'm going to show you an example of texture feature. So this is called gray level roundness matrix, which measures the heterogeneity of the signal intensities in that region. And in this matrix, the ith value would represent a gray level intensity, and the jth value would be the number of connected runs of that gray level. So let's say we have a 2D image array here, and then we can see that there are 3 consecutive runs of a gray level intensity 1, and we indicate that here. So we compute this kind of roundness matrix in various directions in 3D, and then we just average the values together across the direction. And now we briefly talk about how the sparse CCA technique works. So let's say we have 2 modalities, and the rows represent the samples and the columns represent the features. So the goal of sparse CCA is really to re-express those 2 sets of variables such that we get multiple pairs of canonical components that are maximally correlated with each other. So what that means is that the CCA is really able to capture the mutual information between the 2 modalities so that we can have a uniform and homogeneous and comprehensive representation of the 2 modalities. And so each of those canonical component value is calculated by taking a dot product between the sample feature and the canonical weight. And those weights are learned by the algorithm. And here we applied a regularization term to the weight so that the model won't overfit. So eventually we will get multiple pairs of those canonical components, each with a different canonical correlation. So then in our analysis, we just averaged each pair of those components together to get a fused 1D feature representation. So this table shows the results on both training and testing. And the results are evaluated on the Cox regression produced patient risk score. So as you can see, across all the baseline models and the SCCA fused model, the fused model has the best concordance index. And it is the only model that has a hazard ratio confidence interval that doesn't include 1, which is the null hypothesis. And also in log rank, p-value is for evaluating whether the survival distribution is significantly different in the predicted high versus low risk patients. And in this figure, I'm showing you the Kaplan-Meier curves from the SCCA fused model. And as you can see, there's a significant difference in the survival distribution. And we also took the model selected fused features and did a PCA on them and visualized the first 2 components. And we were able to see a decent separation between the predicted high versus low risk groups. And finally, we would like to check like which features are more important in the SCCA fused representation. So then we did that by computing the Spearman correlation between each of the fused features and each of the original radiomic features separately from the T2 and the ADC imaging. And we used FDR to correct the p-values and visualized the radiomic features that have significant correlation with all the fused features. So the larger the correlation, it means the more contribution that original radiomic feature is to the fused representation. And so in this confusion matrix, like the heat map, we show that so the columns represent the feature value of the fused features, and the rows represent the original radiomic features. And then this heat map shows for the T2 imaging, and this heat map shows for the ADC. So the first observation is that seems like the shape features have a lot of contribution to the fused representation, as you can see in this high correlation. And specifically, the shape related to the size of the tumor is important. And also we discovered some other non-shape related features that potentially have a high correlation with the fused feature as well. So these are the texture features I highlighted using the green boxes. And they mostly measure the voxel intensity similarity as well as the voxel heterogeneity. Yeah, and like you can see that they have approximately similar important texture features in both T2 and ADC images. So then to conclude our study, the general takeaway of this talk is that by combining different types of information across different modalities, we might be able to improve our model's performance in both diagnosis and prognosis. So then in our specific study, we found that in the integration of radiomic features from T2 and ADC images improves the model prediction of the biochemical recurrence in prostate cancer patients. And the technique we used, CCA, can leverage the mutual information in those 2 modalities. And it's also an interpretable technique. So then in the future, people can like easily extend our approach to other diseases, and then like I mentioned earlier, in more modalities such as genomic. And actually one important area of research is to correlate the radiomic features that we observe at a phenotype level with the underlying biological basis. So that is done through the genomic or omics features. So some limitations, since we extracted the features in 3D, our results might actually be sensitive to some MRI acquisition parameters, such as slice thickness, which is the resolution in the Z direction. And so in the future, we might be have to do some resampling or simply use 2D radiomic feature to address that limitation. And eventually, we would like to use genomic data to correlate with our radiomic findings. And then we would also like to use a larger cohort. Yeah, so that basically concludes my talk. And I would like to thank my advisor, Dr. Xu, and the members of our lab, as well as the bigger MI group, and also the collaborators and funding sources. Let me know if you have any questions or comments.